Good morning, students. I am Dr. Abhishek Kumar Singh, Tutor, Department of Physiology, MCH Deptt, and I am taking lectures on the topic of central nervous system. And today's topic, in continuation of synapse, is the properties of synapse. Yeah, so we'll start with the properties of synapse. Very important topic. You can get a short note, so even a long question on properties of synapse. The first property of synapse is law of forward conduction. As the name suggests, it is that in synapse, the impulse travel in one direction only, that is from a presynaptic neuron to a postsynaptic neuron. As you can see clearly in the picture, the transmission of the action potential or the impulse is from the presynaptic to the postsynaptic neuron, and this is usually seen in the chemical transmission, though transmission is bidirectional in cases of electrical synapse, which are very spare in central nervous system. The second property of synapse is synaptic delay. This is the second property of synapse. What do you mean by synaptic delay? It is the time which is required for the impulse to be transmitted through the synapse is called the synaptic delay. What is the time which is required? That is the time which is required for the transmission across the synapses, synaptic delay. This time is actually around 0.5 millisecond. And what is this time taken for? We'll again go to another same picture which we have. We'll come to this picture. This is very clearly showing how the time is used during the time of synaptic transmission. What happens first, you can see in the picture, action potential arrives at the axon terminal. Then voltage gated calcium channels open and calcium enters the axon terminal. Then the calcium entry causes the neurotransmitter containing synaptic vesicles to release their content by exocytosis into the synaptic cleft, and then these neurotransmitters diffuse across the synaptic cleft and binds to the ligand-gated ion channels on the postsynaptic membrane. You can very clearly see this is again shown in the second diagram just below the big diagram, how this binding of the neurotransmitter opens the ligand-gated ion channels, this resulting graded potential, the synaptic potential which is produced. This finally results in reuptake of the presynaptic neuron, enzymatic degradation, and then diffusion across the neurotransmitter levels, terminating the signal. So this is all the time which is taken during the transmission of this impulse during the synapse, and this causes synaptic delay, and the time is around 0.5 milliseconds. Then we come to another property. Now what is the significance of this synaptic delay? It helps to find out the number of synapses present in the neuronal pathway. Then the third property of synapse is law of convergence and law of divergence. I'll show you one picture. This picture also shows the law of convergence and divergence. As the name itself suggests, that when many presynaptic neurons converge on one postsynaptic neuron, this is called law of convergence. And when one presynaptic neuron converges on many postsynaptic neurons, this is law of divergence. This is very common in CNS, and it forms the physiological basis for summation and facilitation at synapse. We'll see what is summation and facilitation. One more diagram just to explain this. You can see this diagram, what we have just said, that when many presynaptic neuron is converging on one postsynaptic neuron, that is convergence, and divergence is when one presynaptic neuron converges on many postsynaptic neuron, that is law of divergence. Then we come to the fourth property of synapse, which is IPSP and EPSP. Very broadly or very clearly we have talked about this in the previous class, where we have talked about these two properties. If you want, we can show you that how IPSP is inhibitory postsynaptic transmission, postsynaptic potential, and excitatory postsynaptic potential is the EPSP. So what is EPSP? EPSP is basically the depolarizing potential, is excitatory postsynaptic potential, and which is the hyperpolarizing potential is the inhibitory postsynaptic potential. I can show that in the diagram which we have studied in the last class, which very clearly made you understand this. This was the diagram. You can very clearly see in this diagram, this is local depolarization, that is excitatory postsynaptic potential, and this is the hyperpolarizing postsynaptic potential, which is termed as inhibitory postsynaptic potential. We'll go back to our properties, the fifth property of synapse, and that property is called synaptic inhibition. Synaptic inhibition is of two types. What is synaptic inhibition? This basically serves to maintain homeostasis. It is present to prevent widespread activation of many neurons, and it causes stabilization of neurons by preventing the spread of impulse, and it also prevents neuronal damage. So it has two properties, two importance this synaptic inhibition has. One, it helps in stabilization of neuron by preventing the spread of impulse, and it also prevents the neuronal damage. There are two different types. One is called as indirect inhibition, and second one is called as direct inhibition. We'll see that. This is indirect inhibition, but before that we'll go to direct inhibition. Indirect inhibition you can see here, indirect inhibition is also called as postsynaptic inhibition. That is, it is through formation of inhibitory postsynaptic potential on the postsynaptic neuron. Example to this is, it is seen in some interneurons in the spinal cord that inhibits the antagonist muscle. Neurotransmitter involved here is glycine. It occurs when an inhibitory neuron releasing inhibitory substance act on the postsynaptic neuron, leading to its hyperpolarization. That is, hyperpolarization means inhibitory postsynaptic potential, due to which there is due to opening of chloride channels and potassium channels. So these two channels open, resulting in hyperpolarization, and this hyperpolarization causes inhibitory postsynaptic potential, and this is called as direct inhibition. Example we have seen. It is seen in the interneurons in the spinal cord, which inhibits the antagonist muscle. The common neurotransmitter here involved here is the inhibitory type, example glycine. Then we have another one, which is indirect inhibition. This is indirect inhibition. This indirect inhibition is also called as presynaptic inhibition. The direct inhibition was called as postsynaptic inhibition, but indirect inhibition is also called as presynaptic inhibition. What happens here? And why it is called indirect inhibition? Here the passage of impulse through a synapse is inhibited by another earlier impulse originating from a separate neuron which is terminating on the presynaptic ending. You will understand this with a diagram shown here. Show you one diagram which will explain this. This is the diagram. Usually the synapse which is present here is, you can see here, exo-axonic synapse. So there are two neurons here. What is happening? The passage of impulse through a synapse is inhibited by an earlier impulse originating from a separate neuron. So this is a separate neuron. This is a separate neuron which is there, and this separate neuron releases a neurotransmitter which is usually GABA, and this separate neuron helps in terminating the neuron which is this. You can see here what is happening. Action potential is arriving, number one. Then less calcium is entering, number two. Less neurotransmitter is released, number 3, and then reduced effect is on postsynaptic membrane due to inactivation of calcium channel because of the release of GABA. So this is an indirect type of inhibition or presynaptic inhibition, and usually it is exo-axonic. What is the mechanism which is involved in this presynaptic inhibition? As you can see in the picture, there is decreased calcium entry and decreased release of neurotransmitter. There is voltage-gated opening of potassium channels, and there is direct inhibition of the transmitter release, usually GABA, and this produces the presynaptic inhibition. So we talked about direct inhibition, which was a postsynaptic inhibition, and then indirect inhibition, which is a presynaptic inhibition. These are two types of synaptic inhibitions under properties of synapse. There is another kind of inhibition also, which is called as feedback inhibition and feed-forward inhibition. This is feedback inhibition. Feedback is actually a negative feedback inhibition. An example to this is the Ramshaw cell inhibition in the spinal cord. What happens here? There is inhibition, there is spinal motor neuron ends on an inhibitory interneuron with this inhibitory interneuron. This is the inhibitory interneuron, and this is called as the Ramshaw cell. These inhibit discharge of same motor neuron. So therefore it is called as negative feedback inhibition. The neuron inhibit themselves in feedback fashion. That's why it is called as negative feedback inhibition. Then there is feed-forward inhibition also, about which in detail we'll talk when we'll talk about cerebellum, because it's a very important example is cerebellum, where there is Purkinje cell output which is inhibited through a feed-forward inhibition. We'll talk that when we talk about cerebellum, the part of the brain. Then the sixth property of synapse is synaptic facilitation. As the name very clearly indicates, when a presynaptic axon is stimulated with several consecutive individual stimuli, each evoke a large postsynaptic potential than a previous stimuli, and this is called as facilitation. So the process is by which the transmission through a synapse increase. Facilitation means there is increased transmission through the synapse, and this happens because of the opening of the voltage-gated calcium channels, which remain open for a longer duration. And when these voltage-gated calcium channels remain open for a longer duration, more calcium influx occurs. There is increased transmitter release from the postsynaptic presynaptic ending, and synapse is facilitated. So this property is called synaptic facilitation because of the whole process is, the ideal basis of the whole process is prolonged opening of calcium channel resulting in more calcium influx. Then we come to the seventh property of synapse, and that property is called summation. Summation means add-on. We very well know the word summation. There are two types. One is temporal summation, another is spatial summation. You can see very clearly in the diagram. The name itself will make you understand, the diagram will make you understand what do we mean by this spatial summation and temporal summation. The first is temporal summation. When a single presynaptic knob is stimulated repeatedly, this stimulates the postsynaptic neuron, thus postsynaptic potential is elevated to a firing level. You can see here, a single presynaptic knob, presynaptic terminal is stimulated repeatedly, resulting in production of a postsynaptic potential. And this postsynaptic potential is elevated to a firing level with summation of these multiple stimulus. Then there is spatial summation. This is the diagram showing spatial summation. Here what happens? This occurs when many synaptic nodes converging on one postsynaptic membrane is stimulated simultaneously. These are the presynaptic knobs and these are stimulated simultaneously. These are terminating on one postsynaptic neuron, and when all of these presynaptic knobs terminating are stimulated simultaneously, then there will be two or more separate input which arrive simultaneously at the postsynaptic neuron, and this summation is called as spatial summation. So multiple presynaptic knobs stimulated and they producing an impulse at the postsynaptic neuron is spatial summation. When a single presynaptic knob stimulated simultaneously again and again, producing an impulse resulting in postsynaptic potential is temporal summation. This is another property of synapse, that is summation, regarding which we talked earlier also. Then we come to the eighth property of synapse, and that property is called occlusion. What do you mean by occlusion? What is happening here? Activation of a presynaptic neuron A stimulates a set of 5 postsynaptic neurons, plus activation of a presynaptic neuron B stimulate another set of 5 postsynaptic neurons. But as the two presynaptic neurons share their discharge zone, that is the common area of A and B, then what is happening? Activation of both A and B result in activation of 8 postsynaptic neurons instead of 10, and this is called occlusion. You understood? What is happening here? If A is causing stimulation of 5 postsynaptic neurons, A is a presynaptic neuron, B is causing stimulation of another 5 set of postsynaptic neurons, but when they are both stimulated, A and B both, then activation results there is activation of not 10 but only 8 postsynaptic neurons, because there is sharing, the discharge zone is sharing, there is some common areas of A and B, so there will be, in spite of 10, there will be 8. This is again shown in this picture. You can see here the response of stimulation of B and C together is not as great as the sum of response to stimulation of B and C separately. This is what I was trying to explain. Because B and C both end on a neuron Y. This is the neuron Y. So there is a common discharge zone. This common discharge zone is Y. And because of this decrease in expected response due to presynaptic fiber sharing, postsynaptic neuron is called occlusion. So because of this common sharing of this Y zone, there is this this less stimulation is called occlusion. So there is occlusion of 2. Here, in spite of 10, 5 were, 8 were stimulated. So there was occlusion of 2. So this is the property of occlusion. Then there is something, the 8th property comes as sensitization. What is sensitization? This occurs when transmission is accompanied by a painful or an unpleasant sensory responses, which increases by the mechanism of presynaptic facilitation. We studied about presynaptic facilitation. So sensitization occurs when a stimulus is accompanied by a painful or an unpleasant sensation. Here the response increases by the mechanism of presynaptic facilitation. Then 10th, potentiation. What do you mean by potentiation? Potentiation means enhanced synaptic transmission for a prolonged duration. When there is enhanced synaptic transmission for a prolonged duration, and again this is due to increased calcium entry, this is called potentiation. Now the process of synaptic strengthening is also called as long-term potentiation. When there is synaptic strengthening, this is called as long-term potentiation. Then, so this was the 10th property of synapse. Then coming to the last property of synapse, that is called as synaptic plasticity. I'll show you one diagram which will make you understand this. This is synaptic plasticity, or the plasticity of brain. What do you mean by this synaptic plasticity? Several ways by which synaptic transmission is modified. Modified means either it is weakened or strengthened to suit the need of the body and to learn to react to a particular stimulus. This is synaptic plasticity. Several ways by which a synaptic transmitter is modified, strengthened or weakened to suit the need of the body and to learn to react to a particular stimulus. These changes are memorized by the cells involved in memory and learning. These changes are memorized, and thus this involves a long-term change in the synapse, and this is called as synaptic plasticity. Plasticity refers to ability of the brain to recognize the neural pathways throughout the lifespan as a result of experience. We just talked about this. This is the way the brain remembers the changes, and which is by the cells which are involved in memory and learning. And this long-term change in the synapse is called as synaptic plasticity. So this is changing with the learning. The brain is ability to change with learning. There is a change in the internal structure of neuron, and notably the synapse, and there is an increase in the number of synapse. This all process is the plasticity of the brain. So we just talked about all the properties of synapse, very important from your examination point of view, and you need to understand this, make diagrams which I have shown in the picture to make the teacher understand, to make yourself completely that you understood what you are actually writing in your notes, in your answer sheets. So you have to explain it with the good diagrams, labeled diagrams, and you can explain the properties of synapse. So we talked about 11 properties of synapse. One more thing just before ending, what is synaptic fatigue or phenomena of habituation? When a presynaptic neuron repeatedly is stimulated and a postsynaptic response gradually decline and disappear, this is called as synaptic fatigue or phenomena of habituation. Thank you so much for today. Next class we'll talk about neurotransmitters and we'll again discuss another new chapter in the central nervous system. Thank you, students.